Hello everyone. Today we are going to learn about DNA replication. So DNA replication is going to occur before cell replication. So let's take a look at what it says here. It says that for DNA replication, the cell must replicate the DNA to have two sets for each new cell after cell division. So basically, during the process of cell division, one cell is going to divide into two. You see a picture of that here, right? Each one of these is going to be a new cell that is being created, right? One cell is splitting in half into two. So one of the most important things you can put in a cell is the DNA, right? There's my DNA for each cell. And I need the same DNA in each cell, right? All of your cells have the same DNA in them. So you need enough for each one. So if you've got one cell that you're splitting into two, you've got to divide your DNA. You have to replicate your DNA and divide it up into each new cell in order for cell division to work. So DNA replication is a process that has to happen before cell division. DNA structure really helps in replication. It makes replication pretty easy. And the reason why is that one strand is a template for the other. So if you don't know what I mean by that, take a look at the DNA on the right. And let's pretend like we make one of the strands of DNA disappear. Even if I make that strand disappear, I still know what that strand was, right? And that's because of the complementary base pairing. Right, I know that for that first nucleotide that's a C, it has to be paired with a G, right? One strand tells me the other one. I know that if this is an A, its complementary base pair has to be a T. So even if that strand is gone, even if that strand disappears, I can already predict what it's going to be because of those complementary base pair rulings. So DNA replication has just a few steps. It's really pretty simple. So number one, we've got our DNA. We've isolated the DNA that we want to replicate. And so step two, we have to split up the two strands of DNA. So what kind of bonds are keeping those two DNA strands together? So I'm talking about the bonds right there. What kind of bonds hold those base pairs together? That would be hydrogen bonds. And as we already learned, hydrogen bonds are weak. They're strong, but they're weak, kind of like a Post-it note. So those hydrogen bonds are easy to break. And so the very first step and part of step number two is we have to separate the two strands of DNA as you see in the picture. Separate the two strands. Now, what we also see at the bottom of step two are these new bases, right? These are the monomers of DNA. What do we call the monomers of DNA? The monomers of DNA were nucleotides. And there are four different types of nucleotides, right? We could have A or T or G or C. So those nucleotides that are free and floating around are going to be added into the DNA to build a new strand, right? We see that on step three. Step three, we've added in those free nucleotides to finish the DNA, and now we have two copies of that DNA that we had before. All right, here is another view. All right, here's our DNA, which is about to undergo replication. This is a great picture. We start to break the hydrogen bonds. So those hydrogen bonds are now breaking, and we can see right here is the point at which they're breaking. So the DNA is separating. Now we are adding in new bases for step three. This is the one I'm putting a box around, right? We can see that these bases get added the right way. So if you look, I'm circling that cytosine, that C. That C is probably going to go right there to pair up with the G, and so on, right? All down the line. In step four, we see that we've almost completed synthesizing the new DNA. And in step five, we can see that we have two newly made, and they are identical DNA strands, DNA molecules, I should say. So if you compare those two pieces of DNA, GC, GC, AT, AT, we can see that they are identical. They are the exact same thing. So let's look at each of these steps in a bit more detail. So step one, we've got to unzip or unwind the DNA. You can maybe write that. Unzip and unwind are the same. We need an enzyme to do that. The enzyme we use is helicase, which I am putting a box around. You can remember helicase because helicase undoes the helix structure. So the helicase in the picture is right there. That is the helicase. You can see that it's moving. What direction is helicase moving? It's moving, it looks like, to the left and unzipping the DNA. The point at which this occurs is called the replication fork, right? We call it the replication fork, like a fork in the road. It's where the DNA splits. So the helicase is going to make a replication fork. So that is step one. Step two is going to involve an enzyme called DNA polymerase. DNA polymerase is going to synthesize a new DNA polymer. Polymerase, polymer. So DNA polymerase is going to bind to the chains of DNA that have unwound, and it is going to add in the correct base pairs. So here's a pretty simple question. As we look at this picture, right, we see the DNA polymerase is moving to the right. So what is the next base that's going to be added in? The DNA polymerase is going to add a T with the A. And as it continues to go, it's going to add a G with the C, and a C with the G, and a C again. So the DNA polymerase is going to add in those bases. And here we see a picture of the whole process happening at the same time. We've got the helicase in the middle. Here's our replication fork. The helicase is moving to the left. And then we have our DNA polymerase. Look at it. It's adding in those base pairs. And at the end, we're going to have two brand new molecules of DNA that are identical. Right? There's another picture of DNA replication happening. And here's an interesting view of DNA replication happening, and I would pay attention to this view. Replication happens at what is called, and you should write this down, a replication bubble. Notice how the whole thing is very bubble-like. The replication bubble has two replication forks, one on each end. So it's going in two different directions. It's going to the right and it's going to the left. So there's helicase here going to the right. And then on the other side, there's helicase going to the left. It's happening at the same time. What might be the reason for that? The reason for that is it's going to be much faster to have it happening two times at once. So it's going to speed up the process quite a bit. We refer to this type of replication as semi-conservative replication. Semi-conservative, we'll explain that term in a minute. Basically, semi-conservative refers to replication that ends up with each new DNA being half new and half old. So conservative means to save. So semi is partially saved because it's half old and half new. And then errors, if any errors occur during DNA replication, that is repaired by proofreading enzymes. All right, so semi-conservative replication, we have the old DNA. The old DNA splits into two. We have new DNA being built on each strand, and then we end up with two daughter molecules. Notice that they are half old and half new. So that is semi-conservative replication. And when semi-conservative replication is over, we have two exact copies of the same DNA. And proofreading enzymes are going to proofread the DNA to check for mistakes. If there are any mistakes, the proofreading enzymes will usually repair them so that the DNA is identical to the DNA at the start. All right, and that is the end of DNA replication. let's have a quick question at the end to make sure we understood everything. What are the enzymes of DNA replication, and what do they do? There's 3 of them. Tell me the 3 enzymes we learned and what they do. So first, you should have picked helicase. Helicase is going to unwind the DNA. It's going to break hydrogen bonds and separate the 2 DNA strands. Then there is DNA polymerase. DNA polymerase is going to build the new polymer, so it is going to be making new bonds as it adds in nucleotides. And then we have proofreading enzymes. Proofreading enzymes are going to check for mistakes and correct those mistakes if there are any. All right. That is where we are going to stop for today. That was DNA replication.